Okay, so today we're going to keep talking about muscle. So last time we talked about our somatic nervous system, which we now know innervates skeletal muscle, and it does it by triggering an action potential at the neuromuscular junction. So we have our somatic nerve coming in, releasing acetylcholine at our neuromuscular junction. Then we have excitation, creating an end plate potential on the muscle side. And we talked a little bit about some of the new terms we're going to see when we look at a muscle. So we know we have coatings outside the muscle, then progressively getting further and further in. We have more layers of connective tissue before we hit a muscle fiber itself. It has a plasma membrane called the sarcolemma and cytoplasm called the sarcoplasm. That's where we are right now as we take a look at this structure of a muscle cell or muscle fiber. Okay. So looking inside our multinucleated muscle fiber muscle cell, we're going to see that we see structures called myofibrils, right? So more long threads inside this cell called myofibrils that are surrounded by this structure called the sarcoplasmic reticulum. So that's this purple stuff that you see here. Okay. So we had our sarcolemma as our membrane. We have our sarcoplasm as the cytoplasm. Our sarcoplasmic reticulum, kind of like a modified version of the endoplasmic reticulum that you see in a generic cell diagram. But it has a really special role in a muscle cell, in these muscle fibers. Okay. So our sarcoplasmic reticulum is going to be full of calcium. So it's basically storing calcium ions in these sacs called lateral sacs, right, out to the sides, lateral. Or just a sac in this sarcoplasmic reticulum. We're storing that calcium because ultimately the way we're going to actually contract a muscle is going to rely on releasing calcium into the sac, and calcium is going to kickstart the contraction process. Just like calcium in our neurons triggered that synaptic vesicle to send out its neurotransmitters into a synapse, calcium is now going to trigger our muscles to contract as well. Okay. So that calcium is in the lateral sacs of the sarcoplasmic reticulum. And associated with that sarcoplasmic reticulum, we also have these tubes, right? So I tried to be helpful naming these, right? These are our T tubules. So we're forming a T where we have on the sides, we're thinking baggies of calcium in the lateral sacs, right? And in the center, we've got our T tubule, right? So that's the T we're kind of talking about here, right? Not a great T, but it's looking more like an elephant, but this is our situation. Okay. So here is our triad, which is a T tubule and those two lateral sacs, and they're going to work together to get this muscle fiber releasing calcium in response to action potentials, starting our contraction process. So you can also see how these are located near our motor end plate as well, right? So here's where we have that somatic nerve coming in, right? That's our axon terminal and our terminal bouton. Here we have those synaptic vesicles that would be full of acetylcholine. Here's the motor end plate where we'd have those nicotinic cholinergic receptors, right? So this is where we'd be creating an end plate potential turning into an action potential. We're ready to propagate down this T tubule and interact with this sarcoplasmic reticulum. Okay. So in the myofibrils that are inside this cell, right? So we have this sarcoplasmic reticulum and the T tubules with their calcium getting ready to send signals, but we also have these myofibrils inside in long skinny threads. And those are the things that are going to actually get shorter, right? So our myofibrils are going to shorten the muscle ultimately when we contract. Okay. So the myofibrils are going to be blocked out into units, and within each of those units, we're going to have different parts. And what we want to know first is about two specific proteins that also come in filament form. So, and even smaller thread, right? It's threads full of smaller threads, full of smaller threads when we're thinking about our muscle fibers. So these are our myosin and our actin, right? So here, actin, we refer to as our thin filament because literally it is thinner, right? And our thick filament is this sort of orange peach line in the center. That's our myosin, okay? And they're in these structures called sarcomeres. So when I say there are units to these myofibrils, right, what we're looking at is like, this is a chunk here. So that's going to be one sarcomere. And then we can see there's another chunk next to it there. That's another sarcomere. All right, here's another sarcomere. All right, we're pulling this into pieces. Every time we see one of these little zigzag, what we're going to come to know are Z line. So those are our sarcomeres embedded in this muscle fiber in the myofibril. So here we're zooming in on that myofibril and we're zooming in on a couple of sarcomeres to learn a bit more about their structure. Okay. So when we're thinking about sarcomeres, we have various zones and bands that are named based on how these looked in a microscope slide when we first discovered them. Right, so when we look at the names, some of these names are from what we see here in an actual microscope image. Some link up okay with what we have in our cartoon images in our kind of models, but some of them don't. So we'll point out what those are. I'm going to do these a little bit out of order because I like to think about them kind of from outside to inside, or at least defining our outsides and our middle before we get to what's these squishy zones and bands. So I'm going to point out first our Z line. Okay. So our Z line is going to be like the end of each sarcomere. All right, so here we see a Z line. All right, literally we're writing a Z over and over again, right? And here's our other Z line. So these would be the ends of this sarcomere here, right? So you can also see them in the microscope slide, but they're not so squiggly looking if you were looking at an actual slide in front of you. So they're this thick line here, and this thick line here are Z lines. From Z to Z. The next thing I think is easiest to think about is actually the middle, right? So we're going to think about the M line next. So the M line, we can literally think M for middle, right? It runs down the middle of your sarcomere, right? So here's that M line in the middle, in the center, okay? Now, what I also like about the Z line and the M line is that in addition to giving us our endpoints of the sarcomere versus the middle of the sarcomere, they also both link together one of our types of filaments, okay? So this M line in the center, right, you can see is holding together all these things that look kind of like wheat, right? So those are our thick filaments. That's going to be our myosin later on. So the M line is going to be holding together the myosin. And the Z line is holding together all our thin filaments. So that Z line on the ends, right? Our thin filaments are going to be these like purple blue bits projecting into the neighboring sarcomeres. Okay. So I like those two first because it gives us kind of our boundaries for looking at the sarcomere. And then we can think about the zones and bands. Okay. So when we're looking at these zones and bands, right, we're going to see that we have different gradation of color here in this histological slide. So we can see in what we refer to as our A band, we're going through these two kind of like thick, dark areas all the way to the end of our next kind of thick, dark area here. So that's our A band. It looks dark. And if we link that up to what's happening in the sarcomere, we have it labeled off on this neighboring sarcomere, but we could do it on this one as well. What we're just doing is showing you the full extent of our myosin, of that thick filament. Right? So everywhere you have myosin, it's going to be within that A band. Right? So like the length of the A band would be the length of your thick filament. If you measured it, to get you the same number. That's our A band. Okay. Within the A band, we see our H zone. So we're thinking like writing in H, something like that. Okay. So the H zone is a subset of this. And what it is it's showing us the part of the A band and the part of the myosin where we don't overlap with our thin filament at all. So if you notice, right, like if I were to take this chunk of my sarcomere here, right, I'm circling both part of the thick filament, part of the myosin, and part of the thin filament, part of the actin. Right? So that would be in the A band because it includes myosin, but it wouldn't be in the H zone because we have both types of filaments. The H zone is only this area in the middle where we have just myosin and nothing else. The opposite of the H zone is our I band. Our I band, likewise, is only going to have one type of filament in it. This time, only going to have our thin filament. Yeah. Okay. So you can see that I band, right? So we're drawing an eye like this. Okay. Our I band stretches from one side of the Z line, actually into the neighboring sarcomere, into that other part where we have thin filament but haven't hit myosin yet. So that's our I band. It's lighter in color, you can see here on this slide. So we have a couple of ways of thinking about naming these structures. Okay, so I like to pick Z line and M line as our like endpoints in middle. Then we have our A band as all our myosin, and the H zone and the I band are the parts where we have no overlap. So either just myosin for the H zone or just actin, that thin filament, for our I band. And you're going to practice this, and after you spend some time practicing, hopefully that'll become kind of second nature, but that's kind of the order I recommend thinking about this. We're going to zoom in to the thin filament, to the actin, to the part that's making up those Z lines and then in that I band. It's the only thing in the I band till we get into our overlap area. So our thin filament is actin. It is one of our contractile proteins. Okay? So this is going to be part of what is actually letting you contract a muscle. Now it has layers of structure to it, so we're going to walk through those. So the first thing is we have these little balls or globules. These are our G-actin. So every one of these little balls looks kind of like an olive, has a myosin binding site on it. Right? So what I want you to notice is we're on actin, right? But we have a myosin binding site, right? So what we have on this protein actin is a spot where we grab myosin. Right? So this is going to be the spot where they come together. So you should expect on the actin, we have a myosin binding site. When we get to the myosin, there's going to be a matching spot. Like there's going to be an actin binding site on the myosin so that they can link together. Now, building from a filament, instead of just these little globules, this G-actin, right? We link them together in a line first. That's the F-actin. Think of it like a filament. But ultimately, we're going to have a double strand in this kind of helix structure. So we have a double helix, just like how we do with DNA here, where we have them linked together in lines with their myosin binding sites, all also kind of oriented in a line. So this is going to be important because we have additional kind of regulator molecules that are going to interact with these myosin binding sites and tell us whether we're able to grab hold to myosin or not. So on this diagram at the bottom, what we have is actin, that thin filament, but we also have this tropomyosin. So this sort of spaghetti noodle here, this tropomyosin, is one of our regulator molecules that we'd expect to see in the thin filament. And then we also have this kind of snowman, this troponin complex here. So what we're going to do, if we want to contract a muscle, is we're going to have to move that tropomyosin off of those binding sites, right? So in order to contract our sarcomeres, what we're actually going to be doing is pulling the thin filaments in towards the center of the sarcomere. So we'll be sliding the sarcomere shorter. And to do that, we're going to have to grab onto the myosin, or the myosin is going to have to grab onto that. But we can only do that if these binding sites are exposed. So in the image that we see at the bottom, this is what the thin filament is going to look like when we can't grab onto myosin. So this would be like what this fiber looks like at rest, right? When we're not contracting, we see those sites blocked. And so the tropomyosin is cutting off access to the binding sites. But if we wanted to expose them, what we do is take this troponin complex and kind of use it like a switch, like literally think of a physical light switch, right? We can move its position, right? And it's going to push the tropomyosin. Right? So we have a part of the troponin that's attaching to the actin, right? We have a part that's going to be attaching to the tropomyosin, and we have a part that's going to be attaching to calcium. And you don't have to identify these three pieces specifically for me, but you should know that the troponin complex is doing all three parts. Right? We're holding onto the tropomyosin, we're holding onto the actin, and we've got a part that's going to interact with calcium, telling us if it's time to move the tropomyosin off of these binding sites. So let's take a look at the thick filament now, the myosin. So the thick filament, the myosin, that's what's connected together at the M line in the middle of our sarcomere, right? And it's the myosin and its presence that makes our big A band, right? So anywhere there's myosin, we've got our A band. Then if there's only myosin, we're in that H zone in the middle. So if we look at myosin, we're going to see, well, it's kind of like a golf club, right? So we've got these tails, right? And we've got these heads. Okay. This is myosin. And that tail part is going to be facing in towards the M line, right? So at the middle of the sarcomere. And those heads are out towards the edges. So not at the Z line, but out towards that direction. So in this next picture down, you can maybe see a little bit of a gap here. Right, so this is myosin molecules sitting end to end. And that M line would be here. Okay, so out on this end, we'd have our Z line here, and then the A band where we have just actin, just that thin filament, before we hit our A band and the myosin, and some of our region of overlap. So peeking in at these heads, right? These heads are what's going to interact with our thin filament, interact with our actin. So we can see we've got an actin binding site on the tip of this golf club, or I better call it an oar because we're going to make some rowing analogies later on. So on the end of this oar, we've got our actin binding site. And partway along it, we've got a site where we're going to be able to use ATP, right? Because contracting muscles is going to take energy. So we have an actin binding site and we have an ATPase site. So a site that lets ATP bind, and when it binds, going to act as an enzyme. That's why we have that ACE ending. So that's where we're going to break ATP to use energy. Now, when we see our thick filament from a little further out, if we're not zoomed in just specifically on this protein, we're going to see that we've got lots of them stacked together, right? So we can see lots of heads at kind of different levels here in our thick filament, okay? So we have lots of these heads ready to form, or we're going to refer to as cross bridges. So they're going to bridge across to the actin, right? So that actin binding site, our myosin, will link up with the myosin binding site on the actin. We'll also notice later on, our heads are mostly all over the place, but we do have a center part here called the bare zone where we don't have any heads. We have only tails of the myosin. So right in the middle of our sarcomere, we're going to have just myosin tails, not any of these cross bridges forming. Okay. So the thin filaments and the thick filaments, the actin and the myosin, are really our contractile proteins. So they're the ones that are going to be actively moving. But we do also, we're looking at the kind of full line through the sarcomere, have one more protein. So this is titin. It's the stricken part on the end of the thick filaments. So this isn't made up of myosin. And if we're thinking about the various bands and zones in a sarcomere, I recommend you ignore it. But it does exist, and it does have a kind of important role for the muscle more generally. What it is doing here is just kind of anchoring stuff in place, right? We're going to be sliding these sarcomeres longer and shorter all the time to contract our muscles. So this is making sure they stay aligned right. So that's why we have this kind of spring here, anchoring that thick filament onto the M line in the middle and then that Z line at the end. So this is some structural support. Does literally act kind of like a spring, so it does have this elasticity, this kind of bounciness to it. It's not going to impede us from changing the size of the sarcomere. And now we're going to think about force generation and contraction. So when we think about muscles contracting, we're going to see that the proteins we just discussed, right, the myosin and the actin, they're not actually going to change how long they are. Overall, a muscle is going to get shorter when we contract, right? But the myosin doesn't get shorter, the actin doesn't get shorter, but the sarcomere as a whole does. Right? This is because we're going to see these filaments sliding past each other in what we refer to as the sliding filament model. And I think we have another picture of that in a second. So that's part of the how we shorten sarcomeres, right? So this is why muscles get shorter and why they contract. But then we have this thing called the cross-bridge cycle. And the cross-bridge cycle is a little more granular, so we're zooming in a little more. The cross-bridge cycle is how we're actually generating the force to contract. And so that's where we're going to see the use of ATP. That's where we're going to see calcium coming in and playing a role. So we're going to be thinking specifically about how does the interaction between myosin and actin change over time as we're trying to contract a muscle. And then later on, after we master those two pieces, we're going to think about how do we couple this up to an action potential, right? So how do we get from an electrical signal to forming these cross bridges and sliding our sarcomere shorter? And then later on, we'll be thinking a bit more about where this ATP is actually coming from in a muscle cell specifically. That's kind of our roadmap for the rest of understanding. So we're going to start with the sliding filament model. Okay. So our muscle contraction is actual shortening of muscle. Okay. And the way that this is happening is through sliding rather than actual shrinking of any proteins. What we're changing is how much overlap there is. So I recommend you like take a pen and set it on your desk. I'm going to hold this pen in my mouth. My fingers are the Z line, so this is the actin, the thin filament. This pen is going to be our myosin, right? So what we're trying to understand is how is it that we can make a sarcomere shorter without actually making the myosin or the actin shorter. So all we're doing is this. That's literally it. I know it's a little silly. I do recommend kind of doing it on your desk because people do get tripped up over this somehow. It's probably easier than you're thinking of if you're getting confused on the sliding filament model. Right? All that's happening is that we are sliding these filaments past each other. So when we have a short sarcomere, those filaments are overlapping more. Right? When we have a long relaxed sarcomere, they're overlapping less. And that's it. Okay. So for question, so we've learned parts of the sarcomere. We now know what a long sarcomere looks like, what a short sarcomere looks like conceptually, like what's happening here. So which of those different lines and zones and bands that we went over have changed shape or changed size? And the actin and the myosin themselves didn't, but if we're naming general zones or bands, lines, some of those, if you put them on a microscope side, are going to look shorter when a sarcomere is shorter. Okay, let's talk it through. Okay, great. All right, so we did identify the two big things that are going to happen here, right? So we are going to have overlap changing, right? So when our sarcomere gets shorter, right, we're changing how much overlap there is between thin and thick filaments, right? And so our two structures in the sarcomere that are related to that overlap or not overlap are going to change as well. So the H zone and the I band. Let me see if hold up another picture. Let's draw it out first. And I'll take a look at that picture. Okay. So if we're thinking our sarcomere, right, we're going from Z line to Z line. Right, and we've got our M line in the center, linking up our myosin. I draw a little more actin, a little more thin filament on here, coming off from our Z line. Right. Okay. So when we're looking at this concept of overlap, right, we can see that here we have overlap, right, versus we're looking here, no overlap. And if we're looking here, also no overlap. Right. Okay, so if we make this sarcomere even shorter, right, say we get it so that our actin is almost reaching all the way to the M line in the center, something like this. What we're going to see in a really short sarcomere, right, if we're looking for those parts where we had only one type of filament, right, we've got way less, almost none here, right? And again, way less, almost none out here. So that is your H zone and your I band getting shorter, right? So here, right, that I band was to either side of the Z line, right? It was the part where we had just actin, right? So in a short sarcomere, there's less of it because we have more overlap, right? The overlap part grew. The H zone, where we had only myosin, right? That part is also smaller because it's been taken up by more overlap. So our A band was everywhere we had myosin. Myosin is the same length, so the A band stays the same length. The sarcomere as a whole is shorter because now we have more overlap within that A band. More overlap means H zone and I band are both a little bit smaller than they were before. Now this is happening, this change in amount of overlap is happening because we have the formation and breaking of cross bridges. So we're going to see these paddles on the myosin are going to be essentially grabbing on to the actin and flicking it in towards the middle. So we're going to be grabbing, pulling in order to shorten the sarcomere and shorten it for our contraction. And here are just pictures of what that looks like under a microscope, a relaxed sarcomere, longer, and this shorter contracted sarcomere here with those really noticeably tiny I bands particularly. In the cross-bridge cycle, that's how we're actually taking that actin and moving it in, creating this greater range of overlap and this shorter sarcomere. This is a cyclical connection between the actin and the myosin that needs ATP. So we're going to think about that head on the myosin as our oar paddle. It's going to form a cross bridge. So it's going to bridge across to the actin. You're going to think about that as like your paddle hitting the water. The water in this case is going to be your actin, your thin filament. We're then going to be pulling that oar forward to actually generate force. Okay, so in the cross-bridge cycle, we're going to see steps that are analogous to linking together our thin and thick filament. So when our myosin, our oar, comes into contact with our actin, that's like when we hit the water with our paddle. We're going to have a power stroke where you move the head of the myosin, you move your paddle, which is moving the water, right? So propelling your boat forward, moving the water, moving the actin in order to actually generate force and make the sarcomere shorter. And then we're going to have to do this thing. So it's a cross-bridge cycle because this has to happen a bunch of times to make a meaningful change to the length of your sarcomere to go into the muscle. So you're going to have to break contact with the water. You're going to have to pull your oar out. You're going to have to use some strength to reset to that beginning position, ready to jab your oar back into the water and pull again. This is our goal for our cross-bridge cycle, where we get some of the terminology. Now you pull out in your slide a picture of the cross-bridge cycle, literally a cycle. You've got probably several pictures of it, but at least one. And before we talk through the whole thing, I want you to find for yourselves that first step of the cycle. And I want you to take a look at the myosin and see what's holding on to it at the beginning of the cycle during that first step. Like, other than the myosin, what's in the picture? What's bound to it? Okay, we'll take a look at the cross-bridge cycle right now. So I'll point these out. But yeah, so what you should have seen attached to our myosin, attached to that thick filament at the first step, you're going to see that we're bound onto the actin. We're going to also see that in that ATPase site, the moment we have ADP, adenosine diphosphate, and a phosphate ion linked up. Okay. Let's see what I mean here. So our cross-bridge cycle, right? So we were looking at what's happening right here as we're setting ourselves up for this first step, right? So this peach-ish color, that's our myosin, that's our thick filament. Here's where we're forming this cross bridge with the myosin head. So here's that connection to the actin, right? So the myosin binding site on the actin and the actin binding site on the myosin are touching. And here at our kind of energy site, right, that ATPase site, we can see we have ADP, adenosine diphosphate, and that phosphate ion. So I'm pointing this out now before we actually go through the whole cycle, because it's easy to think that the cross-bridge cycle starts and its first step is when we actually grab on to ATP, our energy source, but that's not the case. That's kind of what happens at the end of the cycle. So when we're setting up our cross-bridge cycle, we're starting our numbering, starting at the point where we already have energy, right? We've already put ourselves in this kind of high energy form. We're set up, ready to pull that actin. Okay? So it's the end part of the cycle that's gonna set that up for our next round. Okay. So first step, right? We're binding our myosin to our actin. And in that ATPase site, we've already harvested some energy, right? So we are in a state where we have ADP and phosphate ion there in the ATPase site. Okay. So when we bind our myosin to our actin, okay, this is then going to trigger the release of that phosphate ion. We don't need it anymore, right? That is just kind of a byproduct of how breaking up ATP works. Right, remember ATP is adenosine triphosphate, tri for 3, right? So 3 phosphate groups. So what's happened at this point already when we're setting ourselves up, we broke off that 3rd phosphate group, right? So that's what that phosphate ion is that we're letting go of. So we release that phosphate ion, taking us into our power stroke, right? So when this phosphate ion comes off, right, we're in a position with our cross bridge of this head, where I can imagine this as like your arm is out kind of straight like this, right? So my arm is the myosin, is this head of the myosin forming the cross bridge. My pen is still my actin here. I got like a straight wrist here. You'll notice as we go into step 2, the power stroke, we fold it in, right? So my wrist, my arm, depending on where you want to put the joint, is bending, right? So we're moving in, right? So that shape change happens in part because that phosphate ion fell off, right? And anytime we're thinking about proteins, when we think about their interactions with other types of molecules, ions, environments, we know that protein shape is really complicated, so changes often create changes in shape. Okay. So we change the shape of this protein as the phosphate ion falls off, and this creates the power stroke where we pull forward or push in, depending on how you want to think about it, right? We take the actin that we grabbed onto as that head changes shape, pulls the actin in towards the center of our sarcomere. Okay. So there's our actin moving inwards towards the center of the sarcomere because this little arm, this cross bridge on the myosin now has that bend in it. Okay. At that point, we're going to empty out our ATPase site, right? So we got rid of the phosphate ion. Now our ADP is going to fall off. that adenosine diphosphate. Right, so we see that ADP getting released, which sends us into our third step. So our third step we call rigor, right? So you want to think of like rigor mortis, right? Like when our body is all like stiff and the muscles are contracted and hard, right? This is rigor for our cross-bridge cycle as well. Our myosin is in a low energy form, meaning we don't have any ATP here, right? And we're in a shape where we can't pull that actin anymore with this cross bridge, right? We already bent in, got nowhere else to go there, right? Our high energy state is when we have our wrists out, right? And have room to flick actin in, right? So this is our low energy myosin in rigor. We're waiting to get more energy to do our next step. Okay. So since our ATPase site is free now, after this rigor step, when we have our myosin in its low energy form, we now have room for a new ATP molecule to bind in to that site on the myosin head. Okay, so for 4, what happens is the ATP binds onto that ATPase site. Now, because there was a change in what's bound to the myosin, it changes shape a little bit, and this ends up making it let go of the actin. So here we see that the myosin and actin have unbound, right? The myosin let go of the actin for right now, right? And it's occupied with this ATP and its ATPase site. So step 5 is we hydrolyze that ATP, hydrolysis, right? We're breaking something apart with water, right? So we break this ATP in half, right? So we break off the phosphate ion, right? And that gives us phosphate and ADP again, just like we saw at the beginning, right? And this cocks that myosin head back into its high energy form. So this is the high energy form that now has the ability to reach out, grab the actin, and flick it back in again. Right, so we've like straightened out this angle that we're ready to grab onto our thin filament again. So this would cycle through, right? So my actin isn't actually just tensized, right? So we've been grabbing it, pulling it in with the power stroke, right? And then we reset and grab further along the thin filament and further along and further along as we slide these two filaments as we study. So looking back at those steps, what was causing the myosin to pull that actin towards the middle of the sarcomere? Okay, yeah, so right immediately before this, right, so part of our power stroke, right, what's actually causing that shape change to the myosin, so let's us do the power stroke that pulls the actin towards the center of the sarcomere, is that release of the phosphate ion, right? So we had that phosphate ion popping off here, right? So it was this piece that causes the shape change in the power stroke, right? So that's the direct cause of this, right? And this hydrolysis earlier on was necessary for getting energy for the cross-bridge cycle as a whole, But this happened a little bit earlier. This happened earlier on in a previous step as we're setting ourselves up to start the cross-bridge cycle. And then the binding of the ATP happens after our power stroke. So spend some time working through that cross-bridge cycle and getting to know the structure of the sarcomere. And when I see you guys next time on Friday, we'll finally get back to that calcium piece and think about how we are turning on cross-bridge cycle. Like, how do we know that it's time to